Shall I start, madam?
Yes, you can.
Okay. Good morning, all. This is Sharada, PGT in Bioscience, Tsrs&Jc Kodakandla, girls. Welcome you all to my online class. Today's topic is structural organization in animals, part 2. Yesterday we have discussed some points regarding that topic. So today we will discuss about symmetry and coelom. So let us start our topic. Okay.
Returning. Okay, so here the recapitulation of a previous day topic. Okay, once again we have to recapitulate our yesterday's topic. So yesterday we have discussed about the level of classification. There is cellular level of classification, tissue level of classification, and organ level of classification, means organization. So what is the example for the cellular level of organization? In case of a cellular level of organization, the example is the nidaria. And tissue level and organ level. Yesterday we have discussed all these things. Today we are going to learn about the body cavity and types of symmetry and type of development, coelom development, and type of true coelom. Okay. Once again, go through this flow chart. So here. So here, kingdom Animalia, level of organization, symmetry, body cavity or coelom, and 5. So here, animals, cellular level of classification or organization will be observed in porifera. Yesterday we discussed these points only. Okay. Now coming to tissue or organ system. So tissue is in case of a coelenterata, and organ platyhelminthes, and organ system. So then onwards means from nematodes to the higher chordates. So all these are the points yesterday we have discussed. Okay. Today we will go for the next topic, that is the symmetry and coelom, which will be useful for organizing the animal. Okay. Now let us come to know about the symmetry. Okay. Now, importance of symmetry. Actually, what is the meaning of it? The concept of symmetry is fundamental in understanding the organization of an animal. Means with the help of symmetry, we can understand the level of organization in an animal. To estimate the organization, otherwise to classify any animal, with the help of symmetry, it will be very easy. Symmetry. The definition for it is the regular arrangement of body parts in a geometrical design related to the axis of the body is called symmetry. Otherwise, symmetry in animals is a balanced in distribution of paired body parts. So how they are distributed and how they are equally divided, and that will be observed in case of symmetry. Okay, let us come to know the types of symmetries in animals. Asymmetry. The animals which cannot cut into two equal parts in any plane are called asymmetrical animals. Here you are observing the sponges. So here we may not able to cut them into equal half parts. That is the reason we are calling them as the asymmetrical animals. Example, sponges. Now let us come to know about the symmetrical animals. The animals which can be cut into two equal half parts in one or more planes passing through the principal axis of body are called symmetrical animals. For example, if we are treated this is an organism, the plane is just like a line. If it is going from the central axis means exactly middle portion. With the help of this plane, if the organism is able to divide into two equal half parts, that will be called as a symmetry. And such type of animals are called as the symmetrical animals. Got it? Once again, so if it is an organism, the central axis is just like the central axis means the plane which is going through the middle portion, middle point, exactly center point of the organism is called as the central axis or principal axis. So such type of a plane is able to cut the or divide the organism into two equal half parts, that will be called as a symmetry, and such type of organisms are called as a symmetrical organisms or animals. Okay. Now, symmetry in animals of two kinds. So usually based upon the features of the symmetry, we are grouping the animals like radial symmetrical animals and bilateral symmetrical animals. Let us come to know first radial symmetry. First of all, we should know what is the definition of it. Okay. When any plane passing through the central axis of the body divides an organism into two identical parts, it is called as radial symmetry. That means just now only we discussed it. So here you are observing the hydra. So in case of hydra, the planes means the lines which are going through the central axis are dividing the organism into two equal half parts. It means here the radial means through any angle. Number of planes are there, 1, 2, 3, 4, 5, 6. Number of radial means a circular manner. We may not give the number for that. So such type of symmetry is called as a radial symmetry. It is the principal symmetry of diploblastic animals such as cnidarians and ctenophores. most probably the diploblastic animals. Yesterday we have discussed about the diploblastic organization. Means if they are having the two germinal layers, diplo, example, cnidarians and ctenophores. So most probably such type of symmetry will be observed in diploblastic animals. Now, the animals with radial symmetry are either sessile or planktonic or sluggish. Most probably they will be sessile, means they will attach to any substance. Okay, and animals showing radial symmetry live in water and they can respond equally to stimuli that arrive from all directions. So here this is very important point. Due to lack of cephalization, means the head region, here we may not able to observe the head portion. Due to lack of that portion, they are responding equally for all directions for any stimuli. And radial symmetrical animals, many planes of symmetry, whereas pentamerous symmetrical animals have the 5 planes of symmetry. Here you are observing starfish. In case of hydra, it is having number of planes for symmetry. But regarding starfish, it is having the 5 planes, which can be able to cut the organism into 2 equal half parts or identical parts. So this is about the radial symmetry. Okay, now bilateral symmetry. So this is another type. In case of bilateral symmetry, when only one plane that passes through the central axis, anterior, posterior axis. Central axis means here you are able to see in the pic. So this is the anterior portion and this is the posterior. The median or sagittal plane. So here you are able to observe it, which is indicating with the blue color. Got it? So the median sagittal plane. Okay. Now, when only one plane that passes through the central axis divides an organism into 2 identical parts called bilateral symmetry. Bilateral means here also we are having the symmetry, but only one plane which is passing from the which type of plane? Central axis divides an organism into 2 identical parts is called bilateral symmetry. Most of the triploblastic animals are having a bilateral symmetry. Bilateral symmetry. Bilateral symmetrical animals are more efficient than the other animals in seeking food and locating mates and in avoiding or escaping from the predators. Because of cephalization, that is a concentration of nerves and sensory cells at the anterior end. This is very important point. So why they are more specific than the remaining ones means here you are able to see the cephalization. Means the concentration of the nerves and sensory cells are locating at the anterior end. Due to that reason, they are leading a better life than the remaining type of symmetrical animals. As a result of cephalization, bilaterally symmetrical animals can sense the new environment into which they enter and respond efficiently and quickly. So usually, due to the presence of head region, they can respond quickly and easily compared with that of the other one. So this is about the bilateral symmetry. Okay. Once again, going through the points. When only one plane that passes through the central axis divides an organism into 2 identical parts. It is called as a bilateral symmetry. Only one plane here. What is that? Median sagittal plane. Most of the triploblastic animals having bilateral symmetry. Bilateral symmetrical animals are more efficient than the other animals in case of seeking food and locating mates and avoiding or escaping from the predators. What is the specific reason for it? Cephalization means the concentration of the nerves and sensory cells at the anterior. Usually any organism, most probably they will have the head region anterior side. So that is the reason the head region is responding quickly. As a result of cephalization, bilaterally symmetrical animals can sense the new environment into which they enter and respond efficiently and quickly. Okay. Now go for the another term. So this is about the symmetry. Now coming to know about the coelom. So I think you heard this word, but you may not have the idea regarding this definition. Now let us come to know. Coelom. Coelom, the term coelom is coined by the scientist Haeckel. The body cavity which is lined by mesoderm is called coelom. Here in this pic you are able to see this is the ectoderm and yellow color portion is the endoderm, and the middle one which is having the red color is known as the coelom. And the coelom is a fluid filled space. The entire portion is filled with fluid, red color portion. The coelom is a fluid filled space between the body wall and visceral organs. Let me explain. What are the visceral organs? The largest organs which are present in our body at the abdominal region are called as visceral organs. And body wall. Body wall means the outer covering area. So here this is the gut and this is the body wall. The gut is covered by endothelium and the body wall is ectoderm. This is the endoderm and this is the ectoderm. In between these two, you are able to see the mesoderm, which is filled with fluid, that is known as the coelomic fluid. And the mesoderm epithelium, otherwise it is called as the peritoneum. The epithelium which is covering the mesoderm is known as the peritoneum. Animals having coelom are called coelomates or eucoelomates. So usually the animals which are having the coelom are called coelomates. So this is about the coelom. The term coelom is coined by scientist Haeckel. The body cavity which is lined by mesoderm is called coelom. Okay. Coelomic fluid, ecto and endo. In between these two, you are able to see the red color portion which is filled with the fluid. That is called as the coelomic fluid. And such type of animals are called as the coelomates. Animals having coelom are called coelomates. And the epithelium which is covering the mesoderm is called as the peritoneum. Okay. Now, acoelomates. Always we mention A for not having, A without coelom. The term itself indicating that they are not having the coelom. That's it. Here, the bilateral animals in which the body cavity is absent are called acoelomates. Let us come to see the picture. This is the ecto. Always outer side is the ecto and inner side is the endo. And the visceral organs inner portion, that is the gut portion. The gut is surrounded by the endoderm and the body wall is having the ectoderm, and in between these two mesoderm is present. But here we are mentioning the term as the acoelomates. Let us come to know why they are called as the acoelomates. In bilateral animals in which the body cavity is absent are called acoelomates. Examples, platyhelminthes. So in these animals, mesenchyme derives from the third layer mesoderm. Mesoderm is present, but it is providing some type of cells which are undifferentiated. Those are called as a mesenchyme, and occupy the entire blastocele between the ectoderm and endoderm. The entire portion of the ecto and endoderm is filled with the mesenchyma. So that the adults neither have the primary body cavity. What is the primary body cavity? Blastocoelom. Or not the secondary cavity. What is the secondary cavity? Coelom. So they are not having the blastocoelom and as well as the secondary cavity coelom. So such type of animals showing as there is no body cavity, the acoelomates exhibit solid body plane. Means due to the presence of such type of mesenchyme cells, that place is solid in structure. That is known as the solid body plane. So this is about the acoelomates. Got it? Once again, I am repeating. Acoelomates in bilateral animals in which the body cavity is absent are called acoelomates. Example is the platyhelminthes. In these animals, the mesenchyme, from where it is derived? From the mesoderm, and occupy the entire gap place of ecto and endoderms. So that is the reason they are not having either the primary body cavity, blastocele, or not the secondary cavity, coelom. So the such type of animals are showing the solid body plane, or we are calling them as acoelomates. Okay. Now, pseudo means not exactly, false, or somewhat. These are the terms we are giving for the pseudo term. Pseudocoelomate bilaterians. Now, in some animals, the body cavity is not lined by mesodermal epithelia. Such animals are called pseudocoelomates. Example, nematodes. Now let us come to know. During the embryonic development, mesoderm occupies only a part. It occupies but only some part of blastocele, where it is occupying adjacent to the ectoderm. So usually ecto is the outer portion and endo is the inner portion. Here mesoderm is there, but it is occupying only the adjacent of the ectoderm region. The unoccupied, which is not occupied portion of the blastocele is called as the pseudocoelom. And pseudocoelom is filled with pseudocoelomic fluid. Okay. Now we are going to learn it about with the help of the picture. Now let us come to know, see the picture. Pseudocoelomates tube within a tube. We are giving a term, tube within a tube. So when you are observing it keenly, how it is looking like such type of coelom? A tube is present within a tube. Okay. Now, as the gut wall is made only ectodermal epithelium. Gut wall is having only endoderm. Okay. And digested food from the lumen of the gut in the surrounding enter easy with the help of a coelomic fluid. Actually, it is the ecto and it is the endo, and this is the pseudo, and this one is the occupied mesodermal region. The pseudocoelomic region is present near the gut wall. So that is the reason it is giving some easy for the transport of material from the gut to the body wall side. Here they are mentioning like that. Pseudocoelomic fluids serve as a hydrostatic skeleton and shock absorber. So due to the unoccupied portion of the mesoderm, such a pseudocoelom portion is useful for the transport of materials. So that is the reason here we are giving a term hydrostatic skeleton or shock absorber. Means it is providing some help for the transport of materials. It allows the free movement of visceral organs and help in the circulation of nutrients and storage of nitrogenous waste. What are the major functions which are done by the pseudocoelom here? It is useful for the transport of material because it is having the fluid-like portion. It is not occupied by the mesoderm. Okay, so it is a mesoderm, the white color portion which is not occupied by the mesodermal cells. So which is having the only fluid portion. It is useful for the transport of materials and as well as storage of waste. So it is giving some help, and that is the reason it is called as the hydrostatic skeleton. Hydro, always the term hydro means water, the fluid, and it is a shock absorber. So here these are the benefits of the pseudocoelomates with the help of pseudocoelomic feature. Means they are having coelom but a little bit, which is not the mesoderm is not covered completely, occupied completely. It is occupied for the ectodermal region side only, not the endodermal region side. Okay. Now, eucoelomate bilaterians. You exactly right, really. All these are the terms we are giving here. The coelom is exactly present here. The coelom or true coelom is a fluid-filled cavity that lies between the body wall and visceral organs. The blue color portion is the ecto. Okay, and here endoderm is indicated with the yellow color portion, and the red color is the mesoderm. The white color portion, which is known as the coelom, means here the mesoderm is having some space that is lined with the epithelium, that is called as the peritoneum. Peritoneum is the epithelium which is covering the mesoderm region. This is about the eucoelomates. The organisms which are having the coelom are eucoelomates. Okay, ecto and endo. In between these two, mesoderm is there, and it is covered both the side with the mesodermal epithelium, that is the peritoneum, and the gap space between these two layers is known as the fluid filled portion, that is known as the true coelomic part or true coelom or eucoelomate. Okay, now coming to mesentery. Just now only we learned that peritoneum is the lining portion of epithelium of mesoderm. A double layered peritoneum that connects some visceral organs to the body wall. Visceral organ means that they are adjacent to the which portion? Endo, and body wall, ecto. A double layer portion will be there. That is called as mesentery. And retroperitoneal organs. Retroperitoneal organs. It is very, very important for 2 marks question in IP exam. Number of time they have asked this question. Retroperitoneal organs. Certain organs such as the kidneys of vertebrates are covered by the parietal peritoneum only on the ventral side. Such peritoneum is called retroperitoneum. Let me explain you 2 terms. The peritoneum which is ectodermal side is the parietal peritoneum, and the peritoneum which is endodermal side is called as the visceral peritoneum. These are the 2 terms you have to remember. Okay, parietal peritoneum and visceral peritoneum. These are the 2 terms. Here the definition for the retroperitoneal organs is certain organs such as the kidneys of the vertebrates. Usually where the kidneys are located in our body? at the dorsal side of the abdominal cavity. In the vertebrates, the kidneys are covered by parietal peritoneum only. Which type of peritoneum? Parietal peritoneum only on their ventral side. Such a peritoneum is called retroperitoneum, and the organs lined by these are called retroperitoneal organs. Okay, two types of peritoneums are there: parietal peritoneum and visceral peritoneum. But in case of certain organisms like vertebrates, the kidneys are covered with only parietal peritoneum on their ventral side. Such peritoneum is called retroperitoneum, and the organs lined by the are called retroperitoneal organs. Now, secondary body cavity. During the embryonic development in eucoelomates, the blastocoel. So yesterday we have discussed about the development of the embryo, morula, blastula, and late blastula. It is having the blastocoel. Okay, the blastocoel is replaced by the true coelom derived from the mesoderm. This is going to happen every time in case of true coelomic animals. So the true coelom is called secondary body cavity. So the coelomic portion which is replaced of the blastocoel is called as a secondary body cavity. As the wall of the gut becomes thicker, because the mesoderm is having the epithelium lining, that is the peritoneum for gut side and as well as the ectoderm side. So that is the reason it become very thicker and muscular. So the digested food and other nutrients cannot diffuse from the lumen of the gut into the coelom. Actually, from the gut, the digested food has to be entered up to the body wall region. But due to this epithelial layers which are covered to the ectoderm side and endoderm side, it is somewhat thick in structure and muscular. So the digested food which has to be shifted from the lumen gut to the outer side is very difficulty for them. Okay, for that region only such type of animals are having the circulatory system. For the transport of such type of material, they are having transport systems or circulatory system. Okay, so secondary body cavity means the cavity which is present between the ecto and endo, which is covered two sides by the mesoderm, is known as the true coelom, and the cavity is known as the secondary body cavity. Otherwise, the blastocoel is replaced by the true coelom, and that is called also secondary body cavity. Okay. Now, Schizocoelomates. Again, the eucoelomates are divided into two types. One is the schizocoelom and second one is the enterocoelom. Let me explain first schizocoelomates. Animals in which the body cavity is formed by splitting of the mesoderm are called schizocoelomates. Examples: Annelida, Arthropoda, Mollusca. And all the schizocoelomates are prostomians and they show holoblastic, spiral and determinate cleavage. Let me explain one by one. Okay, first we go for the pic. Here, in this picture, you are observing here, this one is the ectoderm and this one is the endoderm and this is the mesoderm. And this is a blastopore. This is the blastocoel, because in eucoelomates, we are discussing about the eucoelomates only. Okay, now. So here, The blastocele is having a pore-like structure that is called as a blastopore. And here one more word is there, that is the protostomes. So in case of protostomes, the mouth starts first and then onwards the anus will form. Mouth forms first, then onwards the anus. Okay. So in this picture, you have to observe ectoderm, endoderm, and mesoderm, which is having just like a pouch-like structure. And this is the blastocele, and which is having a pore called as the blastopore. And here we are giving one term, protostome, means the organisms which in which the mouth forms first and then onwards anus are called as the protostomes. Now coming to the points. In annelids, the functional body cavity is the schizocoelom. It is in the form of a series of paired coelomic cavities. So just now only we are observed. So here you are able to see the red color portion just like a cavity of pouch-like cavity, a pair of cavities. And arthropods and molluscans have functional cavity that lies around visceral organs is filled with blood. Okay, so here hemolymph or hemocoel, one by one. In annelids, the functional body cavity is schizocoelom, because here in case of annelids we are observing the paired pouches portions which are filled with coelomic fluid or just look like the paired cavities. That is the reason we are calling them as the body functional body cavity schizocoelom. But in case of arthropods and molluscans, they are having the functional cavities that lies around the visceral organs. Visceral organs means the inner organs with the blood. So that is the reason they are called as the hemocoel. In arthropods, we are calling the coelom as the hemocoel because it is filled with the blood. But in annelids, we are calling it as the schizocoelom because it is having the split of cavities which are in pairing structure. And protostomes means the mouth forms first, then onwards anus. Okay. Now, Enterocoelomates. It is somewhat different with the first one. Now, animals in which the body cavity is formed from the mesodermal pouches of Archenteron. Here also pouches are producing from the mesoderm, but which are attached at the Archenteron. So this is the Archenteron, and these are the pouches formed from that particular portion. Animals in which the body cavity is formed from the mesodermal pouches of Archenteron. Mesodermal pouches, where they are located? At the place of Archenteron. And from that, if the coelom is formed, that is called as the enterocoelom. What are the examples here? They have mentioned here echinoderms, hemichordates, and chordates. In these animals, mesodermal pouches that evaginate from the wall of the Archenteron into blastocele or fuses with each other form the enterocoelom. Enterocoelomates are deuterostomes and they show radial and indeterminate cleavage. So why we are mentioning the term as a deuterostome means first anus will be formed, then onwards mouth. And here we are observing two terms, determinate cleavage and indeterminate cleavage. In the determinate cleavage, if we remove a group of blastomeres at the time of cleavage, there is no chance of formation of the complete organism. Such type of cleavage is known as the determinate. But in enterocoelomates, indeterminate means at the time of cleavage of zygote, the blastomeres, if we isolate from that particular group, even also we are able to get the complete organism. So that is the indeterminate cleavage. So this is about enterocoelomates. Here nothing but animals. Here the body cavity. that is how it is able to form means by the animals in which the body cavity is formed from the mesodermal pouches, which are attached, are formed from the organteron. Here, this is the organteron, and the splitting of the mesodermal pouches into the body, and by the fusion of these pouches, the chance of a coelom is formed. That is known as the enterocoelom. An example: echinoderms, hemichordates, and chordates. These are the examples. And cleavage is an indeterminate cleavage. Okay. Now, comparison. So here with this pic, you can easily understand what are the schizocoelomic formation method and what is the enterocoelomic formation method. So here the examples are molluscans, annelids, arthropods. Here echinoderms and chordates. So here you are able to see the mesoderm pouch and mouth forms first. Here you are able to see the mesoderm pouch, which is forming from the organteron place, and fusion of these pouches cavity is known as the enterocoelomic cavity. And here the anus starts first and then onwards mouth. These are the comparatively differences of both the schizocoelom and enterocoelom. Here also it is a clear pic to write about. If they are asked for the 4 marks question, you can write with the help of this pic completely every point in a detailed manner. Okay. So development of schizocoelom and development of enterocoelom. Entero nothing but organteron, schizo nothing but pouches of the mesodermal pouches. So here you have to write first schizocoelom mouth and then onwards anus. Here you have to write first anus, then onwards mouth. Now go for the next one. In this picture also you are able to see the spiral and radial cleavage and determinate and indeterminate in detailed manner. So this is about the protostomes and this is about the uterostomes. Protostomes means schizocoelomates and uterostomes means enterocoeloms. Spiral and determinate. Spiral means nothing but the cleavage will be look like the spring-like structure. Radial means it is a, you are able to see the cells how they are arranged in a radial manner. And schizocoelous, solid masses of mesoderm split into form the coelom. Here organteron form the coelom. Mouth develops from the blastopore. So usually from the blastopore, what will be formed for the first time? Mouth. But here anus develop from the blastopore. Okay. These are the different differentiations of both the schizo and enterocoeloms. Spiral and determinate cleavage for the protostomes and radial and indeterminate cleavage for the uterostomes. And mesoderm split to form the coelom in schizocoeloms. Organteron form the coelom in enterocoeloms. And mouth develops from the blastopore in schizocoeloms. Anus develops from the blastopore in enterocoeloms. Okay. Now let us come to what we have discussed today. The level of organization, it is a previous day class. Cavity, body cavity, that is today's topic. If it is present, if it is absent. And types of symmetry, radial and bilateral. And coelom, A without coelom, pseudo, light means false coelom, and true coelom. Again the true coelom, enterocoelom and schizocoelom. So these are the points we have discussed today. Let me go for the assignment work. First question for you is a radial symmetry is an advantage to the sessile or slow moving organisms. Justify this statement. I repeat the question once again. Radial symmetry is an advantage to the sessile or slow moving organisms. Justify this statement. Mention the animal. Second question. Mention the animals that exhibited a tube within a tube organization. Second question. Mention the animals that exhibited a tube within a tube organization. Why the true coelom? Third question is why the true coelom is considered as the secondary body cavity. I repeat the question once again. Why the true coelom considered as the secondary body cavity? And fourth question. What are the retroperitoneal organs? What are the retroperitoneal organs? Fifth question. What is coelom? Explain the different types of coelom with suitable examples and neat label diagrams. Fifth question, I repeat once again. What is coelom? Explain the different types of coelom with the suitable examples and a neat label diagram. Okay, I hope you all understood. Thank you.